Polysaccharides have no defined lengths or molecular weights. Additionally, there is no template for synthesis or production of new polysaccharide. This is something we want to keep in mind for DNA. Instead, the program for synthesis is intrinsic to the enzymes that catalyze polymerization of monomer units. So back where we just were, primarily, how do polysaccharides and proteins differ? As you've learned, the answer to the question is the mechanism of assembly. The size of the molecule is somewhat irrelevant to the question, as is the number of carbons present. The mechanism of assembly, however, is totally different because remember that there is no ideal template to follow for a polysaccharide. For proteins, however, they follow the template of DNA to RNA to protein. This process is known as the central dogma and explains how new proteins are produced. Some homopolysaccharides are storage forms of fuel. The two most important storage polysaccharides are starch in plants and glycogen in animal cells. These molecules are heavily hydrated because they have many exposed hydroxyl groups available to the hydrogen bond. These molecules are soluble because of the hydrogen bonding. Starch contains two types of glucose polymers. There's amylose and amylopectin. Amylose is long, unbranched, and connected by alpha 1 to 4 linkages. Amylopectin, however, is even longer with alpha 1 to 4 linkages as well. But is more branched because of the alpha 1 to 6 linkages. Okay, and the alpha 1 to 4 linkages are just the glucose residues. Glycogen is a polymer of alpha 1 to 4 linked glucose subunits. Branches are linked alpha 1 to 6, so it's more compact than starch. And this is just what we wrote out right up here. Okay, glucose is stored as a polymer to avoid high osmolarity. So the question asks, why is avoiding a high osmolarity important? Well, think of what would happen if too much water rushed into a cell. The water in the cell may begin to heat. That's not correct. We're not adding any heat. Osmolarity directly affects the half-life of a cell, maybe in some cases, but not applicable to this question. Thus, the correct answer is A. Too much water may cause the cell to rupture. So, cell wall expands, and the cell ruptures. Next, we're moving on to cellulose and chitin and their presence in the cell walls. Cellulose is tough, fibrous, and water insoluble. It is found in the cell walls of plants. It is linear, unbranched, and a homopolysaccharide. These glucose residues have the beta configuration. Chitin has an acetylated amino group that makes it more hydrophobic and water resistant in comparison to cellulose. So it is even more hydrophobic. And it is a linear homopolysaccharide as well. It is also the second most abundant homopolysaccharide. It is a component of the hard exoskeletons of insects, lobsters, and crabs. It would make sense why they would be hydrophobic. You would not want water permeating the crab. Okay. And in this do you know it question, a microbiologist has discovered an organism in the depths of the Arctic Ocean. In the lab, the biologist has found that the cell wall of the organism was completely hydrophobic and a homopolysaccharide. The organism most likely has a cell wall made of. We know that the answer here is not peptidoglycan. We can eliminate peptidoglycan because it is not a homopolysaccharide. It is a heteropolysaccharide. Which we learned in the beginning of the notes here. Now we're down between cellulose and chitin. And considering that it is in the depths of the Arctic Ocean, we know of organisms existing such as the Arctic crab, we could most likely say that the correct answer to this question is chitin. And plants being down there is a possibility, but we know that the answer is chitin because of the mostly hydrophobic cell wall. Okay. Steric factors and hydrogen bonding influence homopolysaccharide folding. Though in theory there is a rotation about both of the carbon-oxygen bonds linking the residues, steric hindrance by substituents prevents this from happening. So if we were to look at something like this, in theory there could be a rotation. However, likely not due to the limited rotation of steric hindrance. Okay. Polysaccharide have lots of hydroxyl and OH groups. Thus, hydrogen bonding has an influence on their structure as well.